webinars. We have 4 different webinars. There's the recipe book talking about new techniques. We have the sweet and sour with 2 opponents battle it out about different angles of a topic. We have the freshly baked about new publication or recent publication. And as today we have a molecular cuisine, which we always invite a very esteemed speaker to talk about a specific topic and his discoveries throughout his career. And today, my name is Curdin Conrad. I'm here from Lausanne, and I'm having the pleasure to co-chair this kitchen webinar today with Katia Boniface. And she's going to introduce our speaker of today, who's really a living legend, who has formed research over the last 20-25 years and has trained, I think, half of Europe almost, and a lot of researchers who are now in the ESDR actively involved. So Katia, I'll hand over to introduce Tom.
Thank you, Curdin. So it is my pleasure to introduce Professor Thomas Kupper, who is a professor of dermatology at Harvard Medical School and the chair of the Department of Dermatology at Brigham and Women's Hospital. So Tom, you are also the director of the Cutaneous Oncology Disease Center, in Boston. Your research has focused, I think we can say, on 3 main areas. So cancer immunobiology, also T-cell vaccine biology and T-cell immunobiology. And you are worldly renowned for all your work in the identification and the understanding of the role of tissue-resident memory T-cells, so in particular in the skin. And your involvement in dermatology has been constant. So you have been the president of the SID, and you received many awards for all the work that you have been doing with your groups over the year. So we really want to thank you for accepting our invitation to share part of your journey on your work on T-cells and the skin. So the stage is yours.
Well, thank you, Katia, very much for that kind introduction. And this is really a wonderful idea for a lecture series. Let me share my screen. And there we go. Okay, well, it feels like it's been many decades now that I've been studying T cells in the skin. But when I first began on this journey, just beginning to study dermatology and investigative dermatology, we were really looking at skin as almost this this bricks and mortar desert, wonderful barrier tissue, but a relatively acellular, almost like a beach on which inflammatory responses would be washed up and then wash away. And this has altered quite a bit over the last 20 years, I would say. And we're really now at a point where you're looking on FOSS with confocal. This is from Mousse Hanifa. This normal piece of skin contains this bustling marketplace of immune cells, T cells, dendritic cells, macrophages. These cells are all poised to be activated, but very much at rest. And so this is a vast amount of potential energy here. We really stumbled on this observation, I would say, when Dr. Rachel Clark, who's now a professor at Harvard, was a postdoc. We really just looked at explants of skin and found a way to analyze all the cells in this. And in a very simple-minded way, we simply counted the cells microscopically. And done this way, we found that there are roughly 20 billion T cells in skin. So twice as many as there are in blood. Now, this was, at the time, paradigm shifting and surprising, and no one quite believed it, but I think it's quite well accepted now. Not all of these cells are resident cells. We'll talk about that in a little bit, but again, a big observation. And then, this was extended to other tissues, so that the sort of the theme of tissue resident T cells in tissues is they're primarily in barrier tissues, although not absolutely. Skin, lung, and gut. They're at sites of pathogen entry, obviously, unique microbiomes, of course, at all these sites. And they do contain diverse populations of cells. And this idea that that adaptive immunity, at least T cell immunity, grows up in two compartments, both a tissue compartment and a lymph node compartment. So the same naive T cell population can generate cells in skin and lymph node, then gut and lymph node, right on down the road, depending on the pathogen and its site of entry. And so we have these two large compartments of adaptive memory. The tissue element for rapid protective immunity, and the sort of amplifying elements for reserve immunity that can augment this over time. And these play very well together. And these, of course, underline many of the chronic inflammatory diseases we see in skin. But many questions have remained through the years. We've answered a few of these. I'll address a couple of them today. So how do T cells get into skin after infection and become TRMs? What's the molecular process? Are they truly resident? What does that mean, temporally speaking? How well do they truly protect the host against infection? Very importantly, I mentioned these are resting cells. How are they so rapidly activated when they see their antigen? The kinetics are really quite extraordinary. Has a naive T cell become a TRM and not a TCM? Again, that's a binary choice. And how did they survive for so long in skin? Well, I'm not going to tackle all of these. We nearly have enough time, but I'm going to address a couple of them. But first of all, I want to talk a little bit about our model, and that's taking advantage of smallpox virus, or variola major, and an analogous virus, vaccinia virus. Now, this is one of the most lethal diseases in the history of mankind, a true plague that devastated much of the world, and was actually eradicated worldwide by a vaccination campaign in 1977 using a related pox virus called vaccinia. This has turned out to be an outstanding model to track T cell infection of skin. And one of the remarkable things about smallpox is that no one ever gets it twice. This is an old quote from Thucydides. So this is durable, essentially lifelong immunity. So just a word about vaccines. They're delivered in all sorts of different ways. Intramuscular, most common, but also subcutaneous and intradermal. These have always made more sense to me than this. This is a cell-poor network. Skin scarification is really going through the epidermis and into the upper dermis to deliver the payload. And this we found and published. It's a unique way of getting a lot of CD8 memory T cells, tissue resident memory T cells into skin. So I'm going to focus on this, and we used a mouse, very reductionist mouse model to study this. So we're taking a transgenic CD8 T cell called OT1, putting it into a congenic mouse, and then infecting the mouse with a recombinant vaccinia virus expressing OVA, so just this peptide, and then looking at the various memory cells that come out of this reaction. One thing that was found very early on is that this process, and this is from a group in Spain, this process absolutely required the presence of the cross-priming CD8 alpha, CD103 migratory dendritic cell, both in skin and lymph node, and that this was independent of these CD11B high DCs that give direct priming. There is an absolute requirement for this to get TRM precursors. The difference between these two cells is a little bit tricky, and I won't go into that too much, but I want to show you some of our later work. And this is just papers that we've published over the last 15, 20 years, looking at how protective these cells are and how they originate from central or from naive T cells. So I'm going to focus on a pretty recent set of experiments now. One of our big questions has always been, how does this population of naive T cells develop into two completely different lineages from the same environment? So we set up the large single-cell RNA-seq experiment looking at really days 0 through 60 and looking in two compartments, the lymph node and the skin, and done conventional 10x single-cell RNA-seq. This is an enormous experiment, very time-intensive. And this is basically how it's done. We isolate the cells, FACS sort them, and then do scRNA-seq. And even though this looks like a very pure population of cells, there are a fair number of contaminating cells always. So those are filtered out bioinformatically. And we get this this UMAP and these clusters here, and I won't go into this in great detail. These are just some candidate genes. Naive cells cluster separately here, mature central memory cells, mature resident memory cells, and a lot of intermediate lineages, including day 2 activated cells in the lymph node, which are surprisingly homogeneous. This again is just looking at where they are. These 3 clusters are exclusively skin. This cluster is lymph node, obviously naive cells, and these are all intermediary lymph node compartments. And what you can tell over time here is that in the skin compartments, the earliest cells are coming in C2 at about day 5 and 10. But they certainly accumulate much more in the later days. And this is sort of a heat map showing temporally how they're in there. If we do another kind of analysis called trajectory analysis, and ask sort of in what order or sequence do these cells differentiate, what we find is there is a terminal resident memory cluster, cluster 3, and a terminal central memory cluster, cluster 1 down here, shown here, again shown temporally here. And if we apply a technique called absorption probabilities, this bears out very nicely. And again, the genes associated with these sort of polar differentiation states are quite interesting. A lot of immediate early genes and AP1 factors for the TRMs, and much more conventional lineage factors for the TCMs, TCF7, KLF2, and 3. But this caught our eye. And this is simply a temporal map showing that, again, the skin cells start off in the much less differentiated form and come into the terminally differentiated, as do central memory cells here by day 60. If we do something popular like gene set enrichment analysis of clusters 3 and 6, the TRM clusters, we see the usual suspects: TNF alpha signaling, FKB, TGF beta signaling, KRAS signaling. So a lot of pathways are turned up, IL-2, STAT5 signaling. So one thing we were interested in is how unique is skin compared, say, to the GI tract. We looked at a contemporaneously published time course of LCMV virus infection of gut. And if we just look at the skin part of our data and the gut part of Dr. Chang's data, we see this sort of UMAP time course here, where red is late time points, blue is late time points. We find that there is some definite overlap, that there are genes that are markers for resident memory cells, and we characterize these as ones that go up gradually over time. But there are also a whole set of unique genes here. If we look at the heat maps here, they're clearly genes that are going up together, clearly ones that are disparate. Red marks the skin and blue is for the gut. Among these unique genes for skin, there's Icarus going up very nicely in skin and down in gut. This is versus CD103 or integrin alpha E, CCR1 and granzyme C, and there are equivalent genes over here, KLF3, GF1, and ATP8A2. So again, some similarities, but every tissue imprints its own pathways. But one thing that was striking was the AP1 members that we saw in late time point TRMs. And here we're just looking at FOS ligand, FOSB. This is skin and gut mixed together. So it's really almost a linear upregulation of these. Now, at first blush, we thought, well, maybe this is just the digestion we're using of skin and gut. But in fact, the digestion we're using is exactly the same in terms of time course at early time points to late time points. So that's a constant. These are really endogenous to the cells. If we look at regulons, which are a little more sophisticated way of looking at the transcription factors, in FOS, FOSB, FOS ligand 2, Jun B, these are all going up in a linear way in both skin and gut. So fine, this is RNA. Is there any evidence for protein? And if there were, since transcription factors bind to sites on DNA, we should be able to see by a TAC-seq, that is looking at the DNA markers on these cells, whether these are present. And long story short, at day 30, so differentiated resident memory cells, there are all of these open motifs for binding of AP1 factors listed here in sharp contrast. Central memory cells, again from the skin infection, are virtually absent of these transcription factor binding sites. So they have ELK4 ETS, conventional memory binding sites. And what's really striking, and this is done by SciSif immunofluorescence microscopy, is if we look at the skin intact, and this is about 100 days after infection, and we do nuclear staining with Hirsch and Jun B and CFOS, and then put them together, we actually see that these transcriptions of this Jun B CFOS is sitting right here in the nucleus, which is a really striking finding that we are extending and trying to dissect the mechanism of. Now, this sort of harkens back to this innate immune memory concept that Laney Fuchs had some time ago, where in this case, innate cells have this property of partial transcription factor binding to activation sites. So again, the hypothesis here is fairly simple. TCR is bound, calcineurin comes in, NFAT is translocated to nucleus, In the absence of CD28, or in the presence of FOXP3, this leads to T cell tolerance. Immunity, you have the binding of the TCR calcineurin to the nucleus. At the same time, you have binding to CD28 and the formation of this AP1 complex, phosgene. And this super complex is a very, very powerful T cell activation marker for cytokines. So that's what we think may be going on here. We think that the phosgene may already be present in these resident memory cells, meaning that all we need is activation through the TCR, and the cell is now ready to go. And that might help explain the kinetics, the fast kinetics. So let me just in the last 5 or 8 minutes or so talk about yet another tissue, and this is has to do with respiratory viral pathogens, which are certainly a timely subject. Here we're using a modified vaccinia virus anchor that doesn't replicate, so it's the same idea, but not as dangerous to patients. It's a safe vaccine. This has actually been used as the monkeypox vaccine. And we used again our reductionist mouse model. Here again, we're looking at MVA cells that are OT1 or OT1 cells that have the ovapeptide. MVA that has the ovapeptide, we infect the mouse, and then at later time points, we challenge the mouse with a lethal influenza virus, also encoding OVA. So this is an H1N1 virus. The only thing this has in common with this is the ovapeptide. So this is a CD8 T cell mediated protection, if there is any. And indeed, there's striking protection here. We're looking at weight loss and survival. Control mice all succumb. The epidermal disruption, infected mice or skin scarification all survive, and the IMs are intermediate. This is independent of B cells and T cells. CD4 knockouts behave the same way. And the striking thing about this type of vaccination, ED, is if we just look at the lung compartment here, lots and lots of lung CD8 cells here, not seen with the IM. We think that's probably related to this. So we looked at this a little more closely, and we took a mouse that had been immunized or infected with MVA remotely 100 days ago. And then we challenged this mouse. We infected the mouse intranasally with H1N1 flu. And here we're looking at pre-challenge. We're gating on the OT1 cells here. And then if we're looking at these cells, this mouse has been injected with fluorescinated CD45 just before being sacrificed. So this is L-selectin on the vertical axis, CD45 on this axis. Cells that are in vessels will stain positive for CD45. Those in parenchyma will not. So we're just looking at lung, and this is parenchyma, this is vascular, and this is L and positive, L-selectin positive and negative. So only 4 hours after the infection, so before any innate immune response or before any adaptive immune response has happened, it almost appears that these cells have come in from the vasculature to the parenchyma. By 20 hours, they're more abundant. By 3 days, this is even before the central memory cells activated in the mediastinal nodes have come into blood and got to lung. So these cells have really all come from this vascular compartment here. Later on, they're pouring out of the mediastinal nodes and becoming a population of resident memory cells here that will last over time. Now, long resident memory cells are not long-lived, and that's been shown by many, many groups. So our question was, how protective are these cells? They're not resident memory cells. They're not in the parenchyma, but they certainly can get there on innate immune stimulation. So one of the questions we ask is if we can deplete these cells prior to infection, do we lose protection? And then say for contrast, if we deplete somewhere out here, do we retain protection? So we depleted with an anti-CD8 antibody intravenously. And what we found was quite striking. Naive control mice all die. The control OVED mice all survive. If we deplete -1 before infection, we completely lose the protection. We wait just 24 hours, that is just until, you know, these cells have gotten in here, we get very, very significant protective immunity. So we think the lung is probably unable to hold on to too many cells in its parenchyma, but it has this interesting population in the vasculature that are somehow slowed in that compartment. So again, we looked at this by single-cell RNA-seq, here looking at blood in the lung, extrapulmonary blood, spleen, and lymph node. So are these simply just circulating cells? And we hashtag these and analyze them. And here we're looking at different compartments. This is blood, lung, lymph node, and spleen. And then looking how that lays out on a UMAP. There's a lung component with a different transcriptional profile. And here, if we're looking at these clusters, cluster 3 is almost completely in the lung. And if we do heat map, again, looking at cluster 3 here, we find, interestingly, a lot of the usual subjects. We find these AP1 factors, in this case, CD69. So reminiscent of resident memory cells, but not resident. And again, if we look at the hallmark pathways comparing skin, which I just showed you, to lung, they're virtually overlapping. So these cells are we think are really quite new, and how they remain in lung vasculature and whether they're constantly interacting with vessels is something we just don't know. But we think this might be the solution to filling up lung parenchymal space with T cells, which is obviously, from a gas exchange point of view, a very poor adaptive thing. So again, the same idea, if and we haven't shown this yet, but if we can show that our Fos-Jun complexes are in fact already in the nucleus, then simply activation through the T cell receptor might be enough to get these cells to actively engage the pathogen, and we believe that's true from our time course of infection. So let me wind it up there. You know, common themes are these cells are in all tissue, but slightly different themes, slightly different variations on the theme. And just want to acknowledge my very talented lab members here, as well as Chloe Villani and Neil Smith, who helped with the scRNA-seq. And thank you all for your attention.
Thanks a lot, Tom, for an absolutely outstanding talk and some really new data, not just, you know, the coverage of the last 20 years. Just a reminder, you can also put your questions into the chat so we can direct them forward to the speaker. May I start maybe with one? Do you think you've shown now the data on lung, and you have sort of this replenishment of the TRMs potentially through these intravascular TRMs or pre-TRMs? Do you think that the same happens in other tissues? Is this specific for lung? And does it happen in the skin as well, maybe just on a lower level?
Yeah. It's a good question. I think I think it's certainly accentuated in lung, and I you know, keep coming back to this idea that the lung is probably a maladaptive place to build up a population of resident cells. So this may be an adaptation that's certainly accentuated in lung. We're doing some intravital microscopy to look at exactly how these cells are interacting with vessels. But there's some evidence that they're coming in and out, so they're transiting through at a much lower speed. They ultimately transit all the way through, so they are recirculating cells, but they're at any given time there's an abundance of them right there. You know, whether this happens in skin or GI tract, I think at a low level, I think that's probably where the original mechanism originated, but I think it happens at a pace that's almost glacial.
And do you think, I mean, I think it was Dan Kaplan who showed if you remove the tissue memory T cells, you kind of sort of have a circulating pool where you also replenish the TRM pool. So we're all thinking about removing disease-specific TRMs.
Yeah.
So is that feasible? Will it just be replenished from this pool? Or is it anyway not feasible to actually remove the cells in a healthy kind of way?
Yeah, again, the TRM world is sort of gone from there being TRMs and non-TRMs in skin to there being this whole sort of nuanced gradation of those cells, and whether that's the same cells differentiating or de-differentiating, I think that's debatable. I think from a therapeutic standpoint, you'd obviously, in a perfect world, like to eliminate the pathogenic TRMs and keep as many of the protective TRMs as possible. I don't think that's unreasonable. I do think it's like a lot of things in medicine, a threshold effect. I think once you get a certain amount of pathogenic TRMs in skin, that's sufficient to cause disease. So I think if we can lower that number below a certain threshold, I think that's what a lot of biologics are doing. Then the disease simply doesn't become physically manifest.
Yeah, but don't you think in this case after you will need a maintenance therapy? Just in case, do you think we can stop at some point the treatment? That's always
I do think it's going to be a maintenance therapy. The question is, how far can you spread out the maintenance? But I think there will always be some degree of replenishment. Question is, can we slow the pace of that to make the maintenance once every 2 or 3 years?
Okay. So we have a question from Matthias Schmutt in the chat. So is there any evidence for ligands in tissues that may be relevant for modulating the AP1 composition and activation in these experimental models?
No, we're looking at that very carefully. I think we were so surprised to see it, and we're so skeptical for so long that we've looked at all sorts of orthogonal ways to prove this, and I think we're not convinced that it really is there. So I think at this point we're digging into the mechanism and binding partners, but we're not very far along yet.
So I also had a question regarding all the recent work that is coming now regarding the capacity of TRM to recirculate, the identification in human blood of TRM cells. So what is your thought about that? Well,
it's sort of a tautological definition. If it's circulating in the blood, it couldn't have been a TRM, would be one argument. But the other argument is that TRMs are not N-differentiated cells, that whether it's a certain percentage or a certain subset can gain access to tissue and maybe take up residence in another tissue. I think you'd like to argue that for perhaps psoriatic arthritis, it's the same population of TRMs that have found their way to both places, but they obviously got from one side to the other. So I think and with a disease that I study clinically, mycosis fungoides, That's clearly a disease of malignant TRMs, but anyone who has patches and plaques all over their body with the same malignant clone, there's obviously movement of those cells. So how much they have to de-differentiate out of that terminal state and how they reacquire it, I think are all open questions.
Yeah. Maybe I can ask one last question. It's always it's a question also of plasticity of the cells. So do you think, because you have all the work you did and the work with Rachel's group, like where they showed in humans that depending if you take like TCM or TEM, how you can get resident T cells after in the skin. But what I was wondering is whether you would think that at some point maybe a TRM could become like a central memory T cells that would be able to recirculate and then like return to his like fate to become a TRN again, so either like somewhere else in the skin or in another tissue actually.
Yeah, I think we have to make room for that plasticity. I mean, I think that's something that is definitely been observed experimentally. You know, I think it probably has to do with how stable some of these epigenetic modifications are, and yeah, that's again an area that we're just getting into. I think there are a subset of TRMs that are sufficiently modified at the epigenetic level that they're probably incapable of going back. But there are others that maintain some plasticity, and the lung example, I mean, those cells are certainly modified at the epigenetic level, but they're certainly able to transition into lung, become essentially lung resident memory cells, and then de-differentiate and leave. They haven't gone all the way back to being de-differentiated cells. They still have a lot of properties of TRMs, but I think that something sort of analogous to that may be happening in other tissues at this very low level.
Okay.
Maybe a little last question to hook up on that. In this Curdin and Elaine Fuchs showed that particularly also these AP1 transcription factors are in keratinocytes epigenetically altered and have sort of a disease memory that way. Do you think it's the same factors that potentially are altered in T cells, and these are the changes that are seen?
Yeah, I think this is something the adaptive immune system borrowed from the innate immune system, and just linked it up with the TCR as the sensor signal. Yeah, it's not just keratinocytes. There was a nice paper in Nature looking at same thing happening in astrocytes. So I think that's this sort of innate immune memory is a very, very ancient tool for for maintaining longevity.
Right. Thanks a lot.
Thanks again, Tom, for this terrific talk, and so for sharing some new data that you have with your group, and thanks to everyone for attending. So just before we leave this webinar, maybe Thomas, if you can put the slide, thank you very much. Okay. So we wanted to remind you for like the people you knew who were not able to attend that, so you will be able to see soon, so on the YouTube channel, so the video of today's talk of Tom's Kupper. And so we will have the next ESDR Kitchen, which is a freshly baked, so with Juliette Bertrand and Yunsun Chang. So it will be on May 22nd, so still at 1:30 on Wednesday as usual. And so two last information. So the Future Leaders Academy will be in October in Uppsala, Sweden this year. And so please tell all the young promising scientists to apply. So the deadline is soon, it's April 26th. So you can do it through the ESDR website. And lastly, so the next ESDR meeting, as you probably know, will be in Lisbon, so from the 4th to the 7th of September. And so you have until June 3rd to submit your abstract. So the late-breaking abstract will be in July, and you can already register to the meeting. So thanks again to everyone for attending, and looking forward to seeing you next May for the ESDR Kitchen webinar. Goodbye.
Thank you very much.
Thanks
Bye-bye.
again, Tom.
Bye-bye.